Now we talk to the second parameter, relative humidity of the environment. I look for some space to draw. Okay, here first. Now, if for example we put a food on the table here in a cup, we put fresh milk, and here we also have a cup here. We put milk powder. We put on a table overnight, and to the next day, which product will gain weight and which product will lose weight? What do you think? If you leave there for a while, actually the next day you weigh them again. So this one you will lose weight, and this one you will gain weight. What is the reason? The reason here is you have more evaporation of water. Water from the product evaporates to environment. And in the other case, vapor from the air be absorbed into the food more. Here you absorb but less than evaporation. Here you absorb more than the evaporation. And then at the result, you gain weight in this sample, you lose weight in this sample. Which one is planned? Or why? In this case, we lose weight. Why? In this case, we gain weight. Then, this is determined by the difference between water activity of the food and relative humidity of the surrounding air. Water activity of dry milk powder may be 0.45. Water activity of fresh milk 0.99 because a lot of free water. Relative humidity of the air may be 0.75 or 75%, 0.75 in both cases. Here you see that relative humidity here is higher than water activity of the product, so the vapor absorbs more into the product. Here, this one is higher, so the water evaporates more to the environment. Because what is water activity again? Water activity indicates for the vapor pressure. So how easy the water evaporate. If it's high, then it's very easy for the water to evaporate. If it's low, then it's very difficult for the water to evaporate. It will not evaporate, mean the vapor outside will be absorbed inside. Okay, and then what will be the result? This will be the case like I gave you an example already. If your food is not packed very well, then your food will change the water activity. Now, newly absorbed into the food will be quite free. That will be quite free water. So a little bit vapor absorbed into the food will increase quite a lot of the water activity because the newly absorbed water is quite free. And then the water activity of the product increase, means the product will be spoiled. Will be spoiled soon. So then it's important to also understand this principle, this difference, so to do proper packaging for certain products. And we go to the first extrinsic parameter. We go from the bottom to the top. Now we talk about temperature of storage. This is a very important condition, very important parameter because it's influenced strongly to the microbial growth. Now we look at this range of temperature is the body temperature of animal, 37 degrees C. Bacteria multiply very fast. If the temperature is either higher or lower a bit the optimal range, then bacteria will multiply but not fast. If the temperature is further above or below the optimal range, then the bacteria multiply but slowly at a reduced rate, slowly. And then when the temperature is above 63 degrees C and or below 7 degrees C, then the growth will be slow. So this range of temperature from 7 to 63 is called critical range. Remember the word critical range of temperature. in terms of microbial growth. In food products, in food processing, and in food storage, we should avoid this range. Because if we put our food material in this range for a long time, then it's a good range for microorganisms to grow. When we lower the temperature means that we put our food in a fridge or we heat to above 63, then that is already pasteurization, thanh trùng, xử lý nhiệt. Then we already pasteurize the product, we already destroy microorganisms. Here, when we inhibit them to grow, here we destroy them, we kill them. And then this range is critical range, especially during processing. You should not let your food material stay a long time in this range. So if you visit some factory, you will see that the processing room, they have an air gun and they control the temperature from 12 to 16 degrees C. And they control the temperature in the room, in the processing room, like that. So then when during the processing time, microorganisms will not grow very fast. Why they not control at 4 degrees C? Because then it's not friendly to the people who work in the room. It's too cold for people to work. That's why they control at, they condition at 12 to 16 degrees C. So it's suitable for the people who work, it's friendly to the people who work, but also inhibit somehow microbial growth. Okay, so temperature is very important. If we look at one example here, from 12 o'clock, if you have 1 cells of bacteria, now up to 7 p.m., it's become 2 million cell. 2 million cell. From 1 cell up to 7 hours, 2 million. So bacteria multiply very fast. After 20 minutes, each 20 minutes become double in number. So it's increase very fast when the conditions is suitable, like nutrient is available, water activity available, temperature, everything is suitable, then bacteria will grow very fast. And remember one thing again here, that in our food, the food doesn't contain or doesn't contaminate with only one, with 1,000 bacteria, with 1,000,000 bacteria. For example, fresh milk. Food regulation allow fresh milk to have 100,000 CFU per milliliter, mean 10 power 5. So it's allowed that fresh milk has 100,000 or 10 power 5 of CFU per milliliter. It's not only one, it's a lot in the beginning milk already. So if you put milk at suitable condition for bacteria to grow, they will grow in a number of hours. In 4 hours, 6 hours, the milk will spoil for sure. Good. And so when we relate the temperature with microbial growth, we can divide them. We can divide the microorganism into a number of groups. We have psychrophilic group, mesophilic group, thermophilic group, and another group here. What is psychrophilic? Psychrophilic is a group of microorganisms which love to live at low temperature. So the optimum temperature for growth is around 10. They love to live at this condition. And the minimum temperature for them to grow is minus 18. The maximum is 20 only. And because of this, they recommend to do frozen storage at below minus 18. Now minus 20 degrees C, for example. This is the recommended temperature for frozen storage to prevent all the growth of psychrophilic microorganisms to grow, for example. Okay, and then we have a mesophilic, means that microorganisms that love to live at average temperature. And then we have thermophilics, microorganisms which love to live at high temperature. When we do heat treatment, this is the most difficult group to destroy. This is easy to destroy, and this is the easiest to destroy. When you heat a little bit, they already die. Now, but there is an extra group that now we need to know. This is called psychrotrophic microorganism. Psychrotrophic means that this one actually doesn't love to live at low temperature, but it can grow quite fast at low temperature. Even though the optimal range of temperature for growth is up average. They love to live at average temperature, but they still can grow quite fast at low temperature. Means that they can grow when we put our food in a fridge. And this group is called psychrotrophic, and in Vietnamese this is called chịu lạnh. In some previous courses, you already know these three groups, but now you should know more one extra group. It's called psychrotrophic. Ba nhóm này là ưa lạnh, ưa ấm, ưa nhiệt là mình biết rồi đúng không? Ưa lạnh, ưa ấm. I give you an example. We have pasteurized milk. For pasteurized milk, to storage or to do transportation and distribution, we should put in chilling condition or we put in a fridge. We should put pasteurized milk, we store them in a fridge. And then the question is, which group of microorganisms here is the responsible group to cause the spoilage of pasteurized milk in this case? What do you think? The group which mostly causes spoilage of pasteurized milk in a fridge is this one, psychrotrophic group, là cái nhóm chịu lạnh chứ không phải là nhóm ưa lạnh, not this one. Because this is pasteurized milk, means we already applied pasteurization to the milk. When we apply pasteurization, then actually we already kill the psychrophilic because it's very easy to kill them. But when we do pasteurization, part of the psychrotrophic still survive. And then they can still grow in the fridge and they call it spoilage. And also in reality, there will be less psychrophilic bacteria. Maybe you go to the North Pole or the South Pole of the Earth, then there could be some psychrophilic bacteria. But in our country, like in tropical country, then there are not many psychrophilic bacteria actually. Sometimes if you read news, you know that people, they do climbing to the Everest mountain and they die there. But after 20 years, the other people go here and see the intact body because the temperature here is too low. Not so many microorganisms can grow and can spoil the body, can eat the body, so the body is still intact. But for example, in our tropical condition in Vietnam, if some dead animal is here, after several days, it will be very smelly because a lot of microorganisms can grow, can digest the dead body of the animal and generates a lot of smell because the condition in our country is actually favorable to many microorganisms, but not in a very cold weather here. Okay, so if you go to a European country where you walk, normally you go to university, you walk from the dormitory, the student home, to the campus to study. You walk quite like 15 minutes, 20 minutes, but you rarely smell some odor in the environment. But in Vietnam, if you walk from your student home to here, our campus, for example, you can smell many things, many flavor from fish sauce, flavor from a dead animal, flavor from just a trash bin somewhere, because a lot of activity of microorganisms. Good. So we are done with the review. Next meeting, we will go to the main content of the chapter. Do you have questions?